I've been working for the Macroaqua project until 1st of August. Now I switch on for. I'm beginning again, sorry for the repeat. So my name is Delphine Guillebault. I have been working for Macroaqua project till the 1st of last August, and now I switch to run my own company, which partnering with a Chilean company, and we are make available our expertise in macroarray in general sensor. If you want to work with us, you are welcome to meet me and discuss. So now I'm going to introduce the following the next step of the Macroaqua project that Dr. Weigel already introduced to, which concern the criteria for a successful macroarray for identification of pathogen or toxic microorganisms in fresh water. So I will use the Macroaqua project to explain and show you all the steps and how we use all the steps to validate the macroarray of Macroaqua. So I worked in Banyuls with Dr. Guillebault and Professor Linda Medlin. So I will do it very short. It's just as Dr. Weigel said, we cannot proceed with such a huge project which was really ambitious without a good partnership with include 12 European partners across all Europe, and include 6 work packages from the probe design to the management and dissemination. This project has been running over 45 months, and the goal was the development of macroarray for detection of toxic and pathogens microorganisms, including protozoa, bacteria, cyanobacteria viruses, protozoan, and diatoms. So, first step, I would go into more detail than Dr. Weigel did for explaining you what is a macroarray. So, our goal was to elaborate molecular tools to do high throughput analysis of water quality. So, as you know, each individual, like microorganisms, are characterized by a pool of genes, and the genes correspond to sequences. And all the pool of genes and sequences is like a barcode that defines the identity of these individuals. Based on this knowledge, you can find this individual, which is lost in the population, will correspond to one identity, which is a pool of genes. And then when you spot this identity of this individual, like a barcoding, you will be able, in the macroarray, to retrieve from a population a complex mix of microorganisms. You will be able to find back these little ones. So to have an efficient macroarray, you need to have very good genetic markers. So one basic of the macroarrays, which are based on DNA probes, needs to have a good genetic marker and a hierarchy that I define like a barcoding. It means that we will use some genetic markers like the ribosomal RNA genes because they are redundant, they are present in every individual. There's a very good database of the genes, and there's a low gene horizontal transfer from one organism to another one. So when we base all our probes on the ribosomal gene, we are quite sure that the barcoding we are going to define will be unique to an individual. To be sure that the individual is here, we are going to design some probes, which call a hierarchy of probes. This hierarchy will target the groups, the phylums, the class, the genus, and when we can, the species. It means that we will find some unique sequence that will define the groups. For example, when you want to target bacteria, there's some region of the RNA genes, ribosomal RNA genes, that are unique to the kingdom of bacteria. Then when you want to define some probes against the phylum, like the cyanobacteria is a phylum from the kingdom of bacteria, you will define a portion of these genes which will recognize only the cyanobacteria, and so on until you got your target species. it will be a unique set of sequence, and then I call it barcode. Then now I will introduce the critical step of a good microarray. Now, for every experiment, which concerns biology or chemical experiment, you already know that preparation of the sample is crucial. If you want good microarray results, you need to have a good sample preparation. So in the pre-analytic stage, you've got several steps that you will be very careful with. So you will collect your samples, then it will impact on the representativeness of your target. In the project, we developed a standard filtration and concentration process that you saw in the previous presentation, which is really important for material preservation, especially for RNA, which are really, really sensitive to any kind of enzyme. And you need a common extraction protocol to standardize all the material you get at the end. And it gives you high quality results. So good microarray results rely on good material preparation. The second stage, which is really important, is a probe design. Because as I told you, when you've got a good genetic marker, you can do a good hierarchy probe. But the analytical stage doesn't rely only on the probes you design, but also on the quality of the hybridization. You need to be sure that when you will hybridize your material, you will have a good hybridization. So you will include in your experiment an internal control to check for this quality. Then the aim of the microarray project was to identify, so with the probe design, but also to quantify your microorganisms. Then we proceed of the calibration of the fluorescence signal we have on the microarray after the hybridization to correlate it with the cell number. Then, with the fluorescence, we can transform the fluorescence signal into a cell number. Then I summarize on the slides how we process with our environmental samples. So you've got your protocol of preparation of your sample collected from environmental sample. You can also use culture sample. I will show you how. Then you do the total RNA extraction, and all the steps as crucial as the analytical stage. If you do have some troubleshooting with that, you can't go through. It's a go-no-go. Then you've got the fluorescence labeling of RNA, the microarray hybridization, and then the scanning, and you have the signal of the spots which are positive. More the signal is fluorescent, more target there is. So that's the whole process we realized during this microarray project. So the next step to reliable molecular tools is a validation. Because it's good to have a microarray with a lot of probes, but if you don't know if it's specific or not specific to your target, your favorite microorganisms, there's no point developing this technology. So that's what I'm going to present now. So the first step, everything is in silico. You will design your probe. So we didn't redo all the work. We just did a bibliographic survey to check if there's already some probes existing in the literature. And we use PCR probes, fish probes, every kind of probe available. And we did just list all the probes available for each target we wanted to focus on with this project. But the survey bibliography didn't cover all the target organisms we needed, so we had to design some new probes. And to design some new probes, we needed some database, so we constructed a new database for each missing target organism, and we designed the probes from this database. We also modify all the available probes to make them work at the same temperature in the same condition on the microarray. Because this microarray, as you saw in the previous presentation, targets a lot of different microorganisms, and a lot. So there's about 250 probes on the same microarray. And it was really important that all the probes work at the same temperature and in the same condition to give nice result and result we can use. The second step is the validation of this probe, because the in silico work, as you know, is not always reflecting the reality. And when you're putting the microarray at the test in the laboratory, it doesn't give the result you're expecting. So all the targets we have on this microarray followed all these different steps. So we tested all the probes with pure culture of each target microorganisms. We tested the microarray with culture from environmental samples. It means that we took some environmental samples from the same samples and we culture it to have the microorganism presence that we have as a target on the microarray. And then the final validation, we did it with field samples. And we correlate with the cell numbers with alternative methods to validate our calibration curves. So I took as an example the bacteria and cyanobacteria, and we test all this list of microorganisms on the microarray to test the specificity of the probe. It means that for each of those cultures, we individually extracted the RNA, label it, and hybridize it individually on microarray in duplicate. Then, for the specific probe they were on, and the non-specific probe which were not on, were eliminated. Then you've got 3 generations of microarray. The first generation of microarray started with 457 probes. And at the final testing with each culture of pure organisms, we ended up, for example, for bacteria genes and cyanobacteria genes, we deleted a lot of probes. For the generation 2 microarray, some probes were missing to complete the hierarchy of some microorganisms. So we newly designed from the database, we constructed some new probes. And then we tested with pure culture again, until we reached the generation 3 of microarray, and still they were missing some hierarchy probe. And now we've got a generation 3, which is quite complete now. We've got more or less all the hierarchy. And then we've got, for example, for bacteria, 88 specific bacterial probe and 50 specific cyanobacterial probe. This process was long, but now we've got a generation 3 that we can use for fit sample analysis. One objective of the project was to identify and characterize the microorganism, but also to have a quantification of the cell number present, because you're dealing with pathogenic and toxic organisms, so you want to know how many there are, and if there are too much to give the alert or not enough to not to act. So in the same way as the probe design, you need to do the calibration curve for every target. So I will explain you briefly how it works, and I will show you some examples which maybe will help to understand better. So the standardization of all probe signals, when you've got your RNA hybridization, you've got the fluorescence signals, and this fluorescence signals is related to the number of cells. So you extract RNA from a known number of cells, you hybridize on your array, and you measure the fluorescence from this known number of cells.